We will be studying about cardiac output today. In this topic, we will study about what is cardiac output and the methods of measuring cardiac outputs, like indicator dilution method and the other method. So let's start. So first of all, I want to introduce with my notes of cardiac output. Like, so let's start with what is cardiac output. So cardiac output is the quantity of by heart blood delivered by heart to the aorta per minute, and it is a major determinant of oxygen delivery to the tissues. So basically, cardiac output is the quantity of the blood which is delivered by the heart to the aorta. Like we can explain with help of a diagram. Like here is the our heart. Suppose it is four-chambered heart, like this, and almost here we have the aorta. So it is the quantity of blood measured, blood delivered by the heart to the aorta per minute. Okay. Now coming to the next point, a fall in cardiac output may result in low blood pressure. Okay, and reduced tissue oxygenation, acidosis, poor renal failure. Renal failure is like poor kidney functions. Renal is related to kidney. So the next point is, it is the reflection of the myocardial function, and when taken with other measurements such as blood pressure and central venous pressure, the rational treatment of cardiac disorders become clearer. So basically, in this point, we have understood this is that when the patients have cardiac disorders, so we measure cardiac output of the patient along with it is saying other measurements like blood pressure, central venous pressure. It is the pressure in the veins, mainly in the veins. So stroke volume of blood pumped from heart with each beat at rest is in adults is varies between 70 to 100 milligram per ml, while the cardiac output is 4 to 61 per minute. So we can we should learn this for our exams or for our competition material, because it is very important. So the first method is for measuring cardiac output is the indicator dilution method. In this method, it states that if we introduce into or remove something from the bloodstream of the fluid, a known amount of indicator, and measure the of concentration difference of upstream and downstream of the injection site, we can estimate the volume flow of the fluid. So basically, it is saying that if we suppose it's the bloodstream, so if we introduce or if we remove something from our bloodstream or the blood veins, then we can measure the concentration differences from the upstream and the downstream of the injection site where we have injected. Okay, so method employs several different types of indicators. Two generally are employed for introducing the indicator in the bloodstream. That is, it may be injected as a constant rate or as a bolus. It means like if we introduce some indicator, then we can introduce in two methods. The first one is the constant rate. In the constant rate one, we can it means that we introduce the indicator at a constant rate. Constant rate here means we are getting putting some concentration at a specific periods of time into the blood, and bolus means as a whole, like we take the indicator, this amount of indicator, and put the whole indicator in one time only. Now indicator is injected into a large vein, or preferably into the heart's right heart. The right portion of the heart is preferred for the introduction of indicator to measure cardiac output. After passing through the heart, lungs, and left heart, the indicator appears in the arterial circulation. So, what does this point mean? This is very important point. Like suppose we have a heart here, it is four-chambered heart. So, uh, here it says after passing through the right heart. After passing through, suppose it is the right atrium. This is the left atrium. So it is the left right ventricle, and this is the left ventricle. So okay, so it says after passing through the right heart, after passing through the right heart and the left heart, the indicator appears in the atrial circulation. So it goes from the lungs and it goes here. Then here is the aorta. Then it goes through the aorta. So after passing through the aorta or passing from the left part of the heart, it appears in our body, the indicator. So the presence of indicator in the peripheral artery is detected by a suitable photoelectric transducer and is displayed on a chart recorder. So transducer, the photoelectric transducers are based on basically it converts the photo energy or the photoelectric energy. Basically, it catches the photons and it changes the photon energy into the electrical energy so that we can measure the cardiac output. And it is after that we can display it on a chart recorder. So this is a diagram of the cardiac output, concentration and time. Between the concentration and time here, at this point of time we introduced the indicator to blood. After that, here is the maximum concentration reached into the blood. After that, when the concentration is maximum, then it starts to decrease, and then it goes in a non-uniform manner. But at this point of time, we assumed the that our work is done for the cardiac output measurement. Now, the cardiac output, here it says for calculating the cardiac output from the dilution curve. This is the dilution curve. So here it is saying m is the quantity. Take m is the quantity of the injected indicator in milligrams, and q is the cardiac output. So then the cardiac output will be quantity of injected indicator upon average concentration of indicator per liter of blood for duration of curve multiplied by curve duration in seconds. Then the cardiac output will be, if we convert these seconds to minutes, we get a term of 1 by 60, and we can write the cardiac output as q is equal to m into 60 upon area under the curve. Area under the curve here means this. Okay, so here's the indicator dilation method is finished. Now we'll continue to the dye dilution method. Okay, now we will study about dye dilution method. So here from the heading we can say that it is clear that we are introducing a dye into our bloodstream and it is getting diluted, and then we introduce a transducer into our body, and then it will be measuring the cardiac output. So the dye used is in this method is indocyanine green, and it is also known as cardio green dye. And why it is preferred? Because of its property of absorbing light in the at 800 nanometer region of the spectrum, where both reduced and oxygenated blood hemoglobin have the same optical absorption. So here hemoglobin, we have mainly in the blood two types of hemoglobin, the reduced one and the oxygenated one. So in the 800 nanometer region, the optical absorption of the oxygenated and the reduced hemoglobin is 800 nanometer, and the dye which we are using of like the cardio green, it is also having the absorption spectrum of around 800 nanometer. So it is the well suited for our measuring of the cardiac output. The concentration of cardiogreen can be measured with the help of infrared photocell transducer. Like we discussed in the previous topic, and the photoelectric transducers are used. So here we get the more specific type of the photoelectric transducer. Like it catches only the infrared radiations through the photocell, and it will be converting into the electrical energy. So the procedure of this dye dilution method involves injecting a dye into the right atrium. So right atrium, right ventricle, left atrium, and the left ventricle. So we introduced the dye here, and then by means of venous catheter, we actually introduced catheter means a tube. It is a tube-like structure which is used to deliver fluids into the body. So the dye into injecting the dye into the right atrium by means of venous catheter, usually 5 milligrams. Here is the dose of the cardiogreen used in adults, and in children here's the dose, that is 2.5 milligrams. Then a motor driven syringe is used for constantly drawing the blood from the radial and the femoral artery through a cuvette. The curve is traced by a recorder attached to the densitometer. Densitometer is mainly measure the densities. Then after the curve is drawn, an injection of saline is given to flush out the dye from the circulating blood. Like from in the previous topic we have covered that the curve is like this. Then it says after the curve is drawn, an injection of saline is normal, the basic NaCl solution, it is given to flush out the dye from the circulating blood. Now problems related to the use of indocyanine green. So the main problems which are contracted while using the indocyanine green, it has been experimentally determined that above a dye concentration of approximately 20 microgram per ml of blood, the optical density rises less with an increase in the dye concentration that below this level. So here it means that after we reach the concentration of 20 microgram per ml, the change in the graph, the change in the graph is very less. And the second disadvantage is the formation of bubbles in the catheter. And if the bubbles are forming in the catheter, then the cardiac output will be inaccurate. So here comes the completion of our topic of cardiac output. This is the main topics which I have covered from the book of Bioinstrumentation by R.S. Khanpur. And uh, here are some questions which can come into your exams related to the cardiac output. Like explain the detection of cardiac output by a dye dilution method. Here we have to explain the dye dilution method, and then explain the indicator dilution method. We have covered this for the cardiac output. Give properties of the indicator used, like we use the indocyanine 9 or the cardiogreen. We can give the properties of this indicator. Now the question can come like this also: describe the process of cardiac output measurement using the indicator dilution method. Here we have also in here we have to explain the indicator dilution method simply. Then do make the graph in the questions. Here's the very important graph of the cardiac output. Okay, then see you in the next video, and if you like it, if you get some suggestions for me, then do write it in the comment box below. Thank you so much for watching.